Okay, David Roy. David Roy's always here first. Love you, bud.
How you doing today?
Good, good. I've had more than my share of caffeine and we're ready to rock. I often think it's a good thing that like amphetamines are not over the counter because that would be my drug. Be like, yes, let's go.
At least you know yourself.
Yeah, I know. You have to know what to avoid.
So our exam is Monday, correct?
It's 3 days, Monday, Tuesday, Wednesday.
Okay.
Because it's asynchronous, I'm giving people a chance to, get in when they can get in.
Yeah.
A lot of people are working around work requirements, so. And I'm pretty sure we're recording at this point. Yes, we are recording. And I can share my screen. I think I figured out the problem. I had been starting the lecture from Outlook instead of from Teams. And so it was bringing me in as a guest rather than as the host. So I think I have that problem worked out now. Let's hope anyway.
Yeah, I'm also pretty new with Teams. I kind of also prefer Zoom. So
Yeah.
so
I
much
feel
easier.
like Teams is unnecessarily complicated. I mean, Zoom, you just give people a link, they click it, you're in, you record, post it on YouTube, everybody gets a copy. But I don't know. It is what it is. Every university's got their own preferred methods. So
For our exams, are we
10:59.
going to need to really like know in depth of the clinical studies within the chapters, or just like be like kind of
I,
like Oh, go ahead.
I think typically like half the exam, which will be 4 or 5 questions, will be kind of like, here's a problem. What do you think the problem is? And so, like, I may use one of the clinical studies. I think I told you to focus on um,
The sickle
uh,
cell.
anemia thing. So I'll probably ask a question about that. Um, but
Which
yeah,
one is that
yeah.
one?
So I'm sorry.
Which one is that one? Is that not the sickle cell one from chapter 2?
Yeah, yeah,
Okay.
yeah. Tilak's here. Hello, Tilak. I know I'm pronouncing your name wrong. How do you pronounce your first name? I think your mic's off. Yeah, you've got a sketchy connection. You're kind of breaking up a bit. Yeah. Well, anyway, yeah, I can't really hear you. Maybe you can correct my pronunciation at a future date. I've had Indian students before, and I'll know them for years, and I'll be pronouncing their name wrong. And right before they retire, they'll be like, it's Ritika, not Ritika. I'm like, now I know. So don't be afraid to correct me. Patrick's here. Good to see you, Pat. Matthew Tyler.
Good to see you too.
Okay. Some people may show. I think I'm going to go ahead and get started because I usually start with a little bit of review, and if they're late, that's okay. So let me go ahead and share my screen. And remind me if I forget, please. I want to talk about the exam before I close out the show today. So if I don't talk about it, say, hey, wait, you're going to say something. So, okay. I'm going to share my screen. Mm-hmm. Oh, come on, man. Chrome tab, window. Where's my... No, this may take a second. Bum. Again, Microsoft Teams seems to be unnecessarily complicated. Let's see if this works. Oh, window. Why is this not being given as an option? Nope. Okay, there. You see my PowerPoint now?
Yes.
Yes.
Okay, good. And let me know if it doesn't change slides when I think it's changing slides. We had that problem once before. Okay, so reviewing what we did last time, we're talking about muscle contraction. And so we've talked about actin structure, and we talked about different structures involved in non-muscle movement. So now we're getting into sarcomeres and muscle movement. And of course, all movement is based on myosin interacting with actin. I said before, oops, shoot. Where the hell am I? Okay, I said before that, myosin 1 is more structural. This is myosin 2, big long tail, and this is going to be involved in movement. So you've got your head region, which is the globular head. It's got the ATP binding region and the actin binding region. You've got a hinge, which allows it to flex, and the long tail, where the multiple tails line up against each other and they form these thick filaments. So here's your thick filaments. Like right here in the middle, you've got kind of butt end to butt end coming together, and then these go in this direction and these go in this direction. Can you see my
Professor
cursor?
Splish, I'm sorry.
Yeah.
I was just going to say, I can't see the cursor and we can't see the entirety of the slide.
Oh, man. All right, let's stop sharing. Why must this be so hard? Frustration. Okay, can you see this, the myosin thick filament?
Yes.
And you see my cursor moving around?
Yes.
Yes.
Okay, so let's stay in this view and hopefully this will work. So anyway, just back up a tad. We've got the long chain, the long tails of the heavy chains that then line up to form these thick filaments, and they're attaching to each other by non-polar interactions, and that causes them to spontaneously line up and attach to each other. So if you look at the way the heads line up as you go down the thick filament, they're kind of spiraling around. And that's so that they all interact nicely with the thin filaments. Here's a thick filament and 6 thin filaments around it. And here is how the thin filaments are attached attached in the sarcomere. Your thin filaments, as we said, have got cap Z at the plus end, tropomodulin at the minus end, and that's stopping them from depolymerizing. And you've got the troponin complex and the tropomyosin complex, tropomyosin covering the myosin binding sites, troponin controlling the position of that tropomyosin. Here in the Z line, the cap Zs are buried in there, and you've also got intermediary filaments here holding everything together. And you've got our old friend alpha actinin binding microfilaments together. So that's giving you your strength of the Z line to hold everything together. Here we are with our thin filaments again, troponin and tropomodulin controlling its position. Structure of skeletal muscle, we've got many individual cells of muscle. Each cell has got multiple myofibrils within it. And here is your sarcomere, which is the unit of contraction. Sarcomere. So we've got our bands. Again, I'm not a big fan of the band idea, but it's historically what they've done, and It's the way they're going to be asking about it. So it's important to know your bands. So A band is the easiest one. It's just the length of the thick filament, okay? And that's not going to change because your thick filaments aren't changing in length. What's going to change are the areas that are not overlapped. That would be the I band and the A band, or I'm sorry, I just said A band, H band in the middle. So the H in the middle then is thick filaments without thin filaments, and the I is thin filaments without thick filaments. So those are the non-overlap portions, and that's what gets smaller when the cell shrinks. And then we've got these other structural proteins that are important for holding the sarcomere together. Myomasin is holding the middles of the thick filaments in place. And then you've got this guy, titin, holding the thick filaments in place kind of longitudinally here. And then you've got nebulin wrapping around the thin filaments, keeping it straight. Tropomodulin, as we said, at this end, cap Z at this end. Okay, so those are the other structural proteins involved. And here's your contraction. Oh, shoot, this is still wrong. I was still wrong. No, that's right. That's right. This used to say A band, but I fixed it. Okay, so the H band here in the center, it doesn't look like it's getting smaller, but it is. And the I band definitely gets smaller. And you can see that in this video if you want to look at that video again later. Okay, so what is doing the shortening? That's your cross-bridge cycling, which is myosin. It grabs the actin let's see, it lets go of the phosphate, grabs the actin, lets go of the ADP, and it goes to its low energy state, moving the actin. And then it's stuck in that rigor state until ATP comes along, and then it lets go, phosphorylates itself, moves back to its high energy position, and it's ready to let go of phosphate and grab again. So that cycle, that whole idea of the high energy and low energy conformation is kind of important to me, kind of like we talked about that with the sodium potassium ATPase. I like this because it shows you how and why the energy is important in the movement of it. It's really basic biochemistry. You know, you're taking this big, ugly, negative charged phosphate, sticking it where it isn't very comfortable, and that puts it into this unfavorable high energy conformation. And then once it's allowed to come off, it can go back to its low energy conformation. Okay, and here's for the trigger of the contraction, which is controlling the calcium, which is going to bind into the troponin complex. You've got your triad structure, which is the two SRs and a T tubule between them. And your action potential is going to come down that T tubule and interact with the proteins in that triad junction. And those proteins are the ryanodine receptor in the SR membrane and the voltage-dependent calcium channel in the T-tubule membrane. And as I said before, in skeletal muscle, it's really not a channel. It was originally thought to be a channel. I was actually in the lab that helped to isolate this. So it doesn't really act like a channel. It will pass some calcium, but not a lot. Mainly what it's doing is sensing the voltage in the T-tubule membrane. When the cell is polarized, it's in this position and it's plugging that ryanodine receptor. When the cell depolarizes, it goes to this position and calcium can come flooding out. And remember that action potential is like, what, maybe 5 milliseconds? So you can get some really fast contraction relaxation cycles with skeletal muscle, like 1/10 of a second. Or something like that. Okay, motor end plate. This book really seems to like the motor end plate. Talked about it in chapter 2, comes up in chapter 3. I'm getting ready for chapter 8. It's in chapter 8. So our motor end plate then is when the motor neuron comes down. This is a really nice video, by the way. If you haven't seen it, you should see it. The motor neuron comes down and interacts with the muscle in this motor end plate. And the motor neuron will release acetylcholine, which will activate these nicotinic receptors here, and that allows sodium to come in. And once sodium comes in, this area depolarizes. That's the EPP, end plate potential. And if that's great enough to activate sodium channels nearby, you'll get an action potential in the plasma membrane, which will then run down the T-tubules and cause contraction. Oh, and here we are. So there's your acetylcholine, nicotinic receptors. Action potential comes down the T-tubule, And then you get that physical interaction here at the junction, releasing calcium, troponin, tropomyosin, cross-bridge cycling. It's all there. This is really cool. I like this figure because it kind of puts it all together into one spot. Okay. Cardiac EC coupling is different because the cells, like I said the other day with skeletal muscle, you get a stronger contraction by activating more cells, which means more motor units turning on. With cardiac, every cell contracts for every cycle. So the excitation contraction coupling is different here. And the action potential goes cell to cell across the atrium. I'm sorry? Question? Was there a question? By the way, I can't see the chat if you're chatting at me. So just yell out if you've got a question. Okay, so anyway, the action potential is going cell to cell, and it's doing that through gap junctions, which are channels between the cells. So we've got these desmosomes, which are hard spot welds between the cells to keep them together. And then we've got gap junctions, which makes them an electrical syncytium. So that means they all operate together. It's a syncytium. That's a great word, by the way. So anyway, here's your gap junctions. Now here's something interesting I'm going to bring up in chapter 8. I just learned it yesterday. I think it's fabulous. So we know about neurotransmitters. There's such a thing called a gliotransmitter, which we always think of glial cells in the brain as being helper cells, and they're just keeping the neurons alive and healthy and everything. But they also are releasing transmitters, and they call them gliotransmitters. And some of them go out a hemichannel. So a hemichannel is like half of a gap junction. Normally this protein is in the membrane and it attaches to another similar protein from the other side, and that opens a channel between the two and allows electrical coupling between these two. But also I'm finding out glial cells will have just one of them in the membrane. They're not connecting to another cell, and you get regulated release of transmitters through them. Crazy, man. I just learned about that yesterday, and we'll talk about it in chapter 8. Okay, so back to the point though, cardiac EC coupling is about calcium coming in. So when the action potential goes through the cell, comes down the T tubules, we still have T tubules here, the calcium channel here acts as a channel. So when it depolarizes, calcium comes in, and that calcium then activates the ryanodine receptor. So we've got calcium-induced calcium release. So the calcium coming in through the calcium channel is trigger calcium, and then the ryanodine receptor releases activator calcium. And that's what causes the contraction. And then again, it gets pumped back in with the circa. So here's an interesting historical fact that I think tells us a little bit about this anyway. If you've ever done physiological experiments, a lot of times what we're using is Ringer solution, right? Ringer solution is basically all the ions you need to support the activity of the cell plus glucose to keep it going. Usually we've got bicarbonate in there. We're bubbling in CO2 to maintain the pH. Well, Ringer used to use tap water. Like this is like, you know, early 18th century, 20th century, just using tap water to make its solutions. And one day, you know, because scientific standards were getting stronger and higher and higher, everyone switched to distilled water because they didn't realize, but there are a lot of ions in tap water. So they switched to distilled water, and suddenly his hearts wouldn't contract anymore. So he wasn't adding calcium to the solution, but there was plenty of calcium in the tap water. And so that's how he figured out that calcium is important for cardiac contraction. And that's still like a good way to tell the difference between a cardiac and a skeletal muscle cell, because skeletal muscle doesn't need extracellular calcium to contract, and cardiac does. And something else I said in the last class that's kind of important is that the strength of the contraction of the heart is going to be controlled by how much trigger calcium comes in. So this Voltschmetter calcium channel is regulated for how much trigger calcium will come in, which will then control the inotropy of the heart, how strong the heart contracts. So I don't know if we get into that in this class, but I think it's kind of interesting. Well, I mean, it's good for here and now, but the whole inotropy thing, I hope we get into, because, you know, I've trained as a cardiovascular pharmacologist and I really love that stuff. Okay, smooth muscle. Smooth muscle is a lot more complicated in terms of its contraction. They are long spindle-shaped cells, and they've got these little spots all over them, these dense bodies where the contractile proteins kind of interact. Desmin and vimentin, intermediary filaments between them. Alpha actinin, again, holding thin filaments together, but they're not, you don't have that regular sarcomere arrangement, and the myosin thick filaments are not structured when the muscle is relaxing. In a relaxed muscle, your myosin is kind of all wrapped up along itself. When you activate myosin light chain kinase, you phosphorylate these light chains, and then it straightens out and forms thick filaments for the contraction. So another big difference here between skeletal and smooth, and cardiac and smooth, is in skeletal and cardiac, it's really only the actin fibers that are being regulated by that tropomodulin. Excuse me. But in smooth muscle, both the thick and thin filaments are being modulated. So anyway, this aspect up here, this signal transduction, we're going to talk about in chapter 8 and 9. So don't worry about that. But you get production of IP3, which releases calcium. Now from here, this is what's important for this test. Calcium comes out of the SR, binds to calmodulin. Calmodulin binds to myosin light chain kinase and activates it. Myosin light chain kinase phosphorylates the light chains, and then the thick filaments form and they're ready to go. On the other side, on the thin filament side, you've got caldesmin, which is acting kind of like tropomyosin in that it's blocking the myosin sites. So calmodulin, when it binds calcium, will also bind to caldesmin and pull it off the thin filaments, allowing the thick and thin to get together. And another way to get there is activating protein kinase C, which will phosphorylate caldesmin, and again, allowing the myosin to interact. So I think we went over this too. Here's your basic structure of the spectrin membrane skeleton. I like this better, but whatever. I mean, here's glycophorin, which is a transmembrane protein, and that's anchoring the structure to the membrane. Here in the center, we've got band 4.1, adenosine, and a short actin protofilament here. And again, tropomodulin binding the minus end and blocking it up, so it doesn't depolymerize. And then the pink and blue are spectrin. So we've just got an alpha and a beta strand for that spectrin. And it goes from the glycophorin over to this. This is a band 3 protein, another transmembrane protein. And what's interesting about this for me is it actually has a functional role as well as this structural role. So structurally, it's holding the skeleton to the membrane. Functionally, it's a chloride bicarbonate antiporter. And chirin then helps to bind all of this to that band 3. And this is just a repeat of this over there. So the thing repeats over and over again on the membrane. And this, I believe, is like the cover of this textbook. That's how much they like this structure. So there's the glycophorin and all of that, and then the spectrin coming out here, and then here's your chloride transporter, chloride bicarbonate transporter. Okay, and then we looked at this, which I think is really cool. We'll look at more of it today. All right, did I talk about intermediary filaments last time, or am I starting here? Anybody know?
I think we're starting here this time.
Okay, great. So intermediary filaments, I think of the three, are probably the least interesting. They're only structural, which to me makes them the least interesting. There's no movement involved with them. But anyway, this table kind of shows you the difference between your microfilaments and your microtubules versus intermediary filaments. Your actin and tubulin are the same in all cells. They're small globular monomers that you polymerize into these strands. And with your intermediary filaments, and this is probably what's most important about them, they are heterogeneous. There are different intermediary filaments in different cells. And the reason I say that's interesting is because, you know, if you have a metastatic tumor, the way you figure out where that tumor started is you look at what intermediary filaments it has. And that's going to help you with your diagnosis and treatment of that particular cancer. And these intermediary filaments are fibrous, which means they're not small polymerizing out. They're already pretty big, and they polymerize together. Your microfilaments and microtubules, you've got pools of these monomers in the cell because they change often, and those pools are ready to do that changing quickly. Your intermediary filaments are not so changeable, I guess. Basically, all the monomers are incorporated into fibers, and they're not like going to be changing a whole lot during the function of the cell. To put them together, your microfilaments need ATP, your microtubules, which we'll talk about in a minute, need GTP. These do not need any energy. They just spontaneously will polymerize. And another important thing, really important thing about our microfilaments and microtubules is they've got a directionality. You've got a plus end and a minus end. And that's really important for movement in both cases. Here with the intermediate filaments, there's no polarity to it. It's just a structural fiber that's holding stuff together. And here's, what's this? Oh, don't memorize this. I just want to throw this out here as examples of different intermediary filaments. You know, you've got keratin that's involved, that's in a lot of endothelial cells, neurofilaments and neurons, vimentin filaments, okay, nuclear lamina. This is the one that I will talk about, because when we get into cell division, mitosis, the nuclear lamins are going to be regulated. So, you know, you've got a nuclear membrane. If you want to separate the chromosomes, you need to get rid of the nucleus. And the nuclear lamina are holding the nuclear membrane together, holding that structure. During mitosis, they'll depolymerize, and then the nucleus falls apart. And you can separate the chromosomes, and then they repolymerize and you make nuclei again. So this is one we'll talk about later, 2 tests from now, I think. But anyway, other than that, they're just kind of structural and they're tissue specific. That's mainly what you need to know about this. And here's how they polymerize. This is a monomer, and the monomers will coil around each other. So you got a coiled coil dimer. Remember, that's just a structural thing that happens a lot because it makes it stiff. That coiled coiling. And you take that coiled dimer and you overlap it with another one. And now they're staggered here. So as they're overlapping, they're kind of almost nucleating more polymerization. So here's that with the staggeredness. And now this one comes in where it's hard to see where one begins and one ends. Here's kind of a junction here. So you're overlapping, and that's keeping that structure together. So they're going to polymerize out that way. And a whole bunch of them will polymerize together and form this strand, which then winds around itself, again, like a rope, many strands wrapping around each other for strength. So that's about all you need to know about intermediary filaments. They're wrapping themselves up and they're being used for structure. Okay, questions about IF? Nothing? Okay, good. So, last thing to talk about, microtubules. So as we said at the beginning of this lecture, you've got 3 different size structural proteins here. Microfilaments, which are the thinnest, intermediary, which are in the middle, obviously, and then microtubules, which are the biggest structural elements here. So these microtubules are made of tubulin. So tubulin's a heterodimer with a beta subunit and an alpha subunit. And that's important because when they start to polymerize, you're going to have polarity here, just like we did with microfilaments. You've got a plus end where the beta is sticking out and a minus end where the alpha is sticking out. And this is more or less the structure. If you look down the barrel, you've got 13 protofilaments in a circle. And you've got a nucleation site. I don't like this figure. You don't see the nucleation as well as I would like to show it. Let's see, what is the next figure? Yeah, the next figure shows it better. Oh, this is one's from your book. Here's the one from the other textbook I use. So here you can see, as you add monomers to this microtubule, it kind of spirals out. So, and the important thing about that for me is that shows you just the importance of nucleation. You know, if it was flat on top, then adding monomers would be more difficult. But here, because you've got two monomers sticking out, the next monomer will come onto it, and the next onto that, and onto that, and onto that, and it polymerizes out, spiraling away. And an important thing to know about that polymerization too, I don't have a figure about it, but basically your alpha and beta tubulin each have a binding site for GTP. So microfilaments ATP, microtubules GTP. So if you put GTP on these, then they're kind of activated, and now they can be added and allow polymerization. So stop party. So to polymerize, you need GTP added to that heterodimer. Now, once it's in the structure, it's going to hydrolyze that GTP to GDP, and that's going to destabilize the microtubule. So just like microfilaments, as they polymerize out, when they get as long as the cell needs them, you need to cap both ends, or else, you know, they hydrolyze their GTP and they depolymerize. And in microtubules, that's called catastrophe, when they fall apart, because they don't depolymerize one bead at a time, they just boom, fall apart. And you'll see a little animation of that a little bit later. So keep in mind here, again, because I don't have a slide on it, our polymerization is dependent on GTP being there. If all your monomers have GDP, they don't polymerize. And if you stop adding GTP, like if it's an in vitro experiment, and the microtubules will spontaneously polymerize, but when they run out of GTP, they'll also spontaneously fall apart. So it's very GTP dependent. Okay, so this is a single microtubule, 13 protofilaments around. They can also be in doublets. So that, like, you've got an A tubule that's got 13 and a B tubule that's only got 11. That kind of shares two of the A's. And then you've got triplets like this. So this is mostly what we see in the cytoplasm, and it's mostly what we're going to see in the mitotic spindle. These are found in the cilia and flagella, so they're called axonemal. And axonemal just means cilia and flagella. And then you've got your centrioles, which are going to be kind of the nucleation site of our microtubules. And these centrioles have got triplet microtubules in them. Here's a centriole picture here. You can see 1, 2, 3, 4, 5, 6, 7, 8. It's 8 or 9. I lost track. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplet microtubules in a circle. And it forms that kind of barrel shape we see with centrioles and microtubule organizing centers. So we'll come back to this in a minute. But basically here, think about your polarization, and you've got 3 different structures you can make these microtubules into. Okay, so here's our centrioles. If you look kind of through the green into the center of this structure, you can see these 2 barrels, one this way and one that way. That's your centrosome. So, I mean, that plus these proteins are your centrosome. So the centrosome is composed of those microtubules plus all this green stuff around it, which is paracentriolar material, which is a great way of saying something around the centrioles. Peri means around, so paracentriolar material around it. And that pericentriolar material then is going to provide nucleation sites for the polymerization of microtubules. Just, you know, that's the rate limiting step of making a microtubule. You need to have that that nucleation site to start from. So in this paracentriolar material, we've got those nucleation sites, and the they'll start polymerizing from there out. Important thing to know about that is your minus ends will always be embedded in one of these centrosomes. And so that's why we call this the microtubule organizing center, because all the microtubule will be organized from this. And the outer ends are all positive and the inner ends are all negative. And that directionality is key. We'll see how that's used in the cell in a minute. What's this guy saying? I think this guy is saying how you can polymerize up, you can polymerize and get longer or shorter by depolymerizing from that plus end. The minus end is being blocked or stabilized, so you're not going to change at the minus end. Not entirely true, but more or less true. The reason I say not entirely true is some studies show that during mitosis, there'll be pulling on the minus end of a microtubule and depolymerizing as opposed towards it. We'll get to that when we do cell division, but for the most part, think about that minus end being stabilized and not depolymerizing. Okay, here's our centrosome again. This is just a different picture from a different textbook. Oh, shoot. Here is a transmission electron microscopic view of it. You can see one barrel shooting out at you, the other one off to the side, and paracentriolar material around it. And this is kind of, this is in vitro, but this is kind of how it was going to work in the cell. You've got the centrosome in the middle and everything's shooting out away from it in every direction. Okay, so again, this is what the nucleation site is. So this paracentriolar material is made up of gamma tubulin and gamma tubulin ring proteins. So these two together make up what's called a gamma Turk. So your gamma Turk then is these blue monomers here, and the green monomers. So the green monomers here are the gamma tubulin, and the blue and the brown are part of the ring proteins that are holding the tubulin. So what you can see here, and this is kind of, I really love this picture. It just makes it so much easier to see. You've kind of got a situation where you've got this nucleation site right here. So with this nucleation site, you just put a tubulin monomer there, and then another one, and it's going to spiral as it polymerizes out from that site. So key thing here, your gamma Turks are the nucleation sites in that paracentriolar material, and that's why everything starts from here, because you've got that nucleation site. Okay, is there anything else I want to say about that before we move on? Okay, so any questions about that? We all good? Okay, so just to move on here, just like in microfilaments where we had ARPs, here we've got MAPs. These are microtubule associated proteins. They're going to be involved in regulating the microtubules and bundling them and all the things that we saw ARPs doing. We've got MAPs that do that here for microtubules. So here are two examples of bundling proteins. This is MAP2, which is going to make looser bundles. You can see it's a little bit longer. And here's the tau protein. Should have called it the omega protein, maybe. The tau protein, which is going to make tighter bundles. You can see those tighter bundles here. So this would be like a really tight microtubule bundle that goes up the center of an axon. So you've got some stiffness there, so it's not going to break apart easily. And that gives them a nice tight structure. And remember, well, you probably know this. If you don't, I'm throwing it out here. Tau can be a problem in the brain. If it starts to polymerize with itself, it forms neurofibrillary tangles, which is what leads to Alzheimer's, or it's thought to lead to Alzheimer's. They're still not 100% sure what causes Alzheimer's, but this is one of the things that they see in Alzheimer's, which is tau is bundling up to get not with microtubules, but with each other and forming a mess in the brain. Okay, but anyway, what we need to know is tau is making a nice tight bundle here. And then we've got other proteins that are going to block the plus end. So EB1 here is a plus tip protein. It's going to bind here and stop it from depolymerizing when GTP goes away. And then we've got MCAC. MCAC is kind of like cofilin. It's going to depolymerize from one end, help to depolymerize. We've got something like gelsolin as well. Catenin. It's going to sever in the middle. And again, once you sever in the middle, you can depolymerize at 2 plus ends, right? You're giving yourself more plus ends to depolymerize. And similar to our thymosin beta 4, we've got stathmin, which is going to sequester the monomers so they don't, spontaneously polymerize. So we've got all the similar kind of proteins here that we see in microfilaments. And here is your structure in the interphase cell. So your normal cell is just doing its job. You've got the centrosome is centrally located and sending out microtubules in every direction. And the key to that, the reason for that is these microtubules are going to be used as tracks to move vesicles around. So any protein that is either going to be secreted or a membrane protein will be made in the rough ER. And once it's made in the rough ER to be packaged into a vesicle, and a motor will be put onto it, the motor will be put on a track, and that will drag the vesicle in a direction. This over here is mainly here just to show you that if you're going to go into research instead of medicine, you should definitely do immunocytochemistry. I mean, look at that. Isn't that gorgeous? You're staining the nucleus in blue and the microfilaments in green, and you can just see how they start here at the MTOC and then gorgeous, gorgeous, gorgeous. Okay, so the other type of map is the motor map. The maps above we call non-motor maps because they are not motors. Down below here, they are motor maps. So we've got 2 different motor maps. And the reason we've got 2 is one goes from minus to plus and the other goes from plus to minus. So kinesin, I always think kinetic movement goes towards the plus end. That's the way I memorize it. So kinesin is then a motor. You can see it looks very similar to myosin, and it will go from the minus end to the plus end. So at one end of this kinesin, you've got, similar to the globular head of the myosin, you've got ATP binding and hydrolysis, and also binding to the microtubule. And then you've got the long structural coiled coil in the middle, and then you've got proteins that are going to connect it to the cargo vesicle. I'll show you that in a second. Very similar to what we see with the spectrin spectrin skeleton. So that is your kinesin. Now, dynein is structurally very different from kinesin, but it does the same thing. It's just a weird looking structure. So it's got one head that's going to bind to the microtubule, and then the business part is in here. It uses ATP and changes conformation. So that's your dynein. So as I said, your kinesin is going to move towards the plus end, and dynein is going to go to the minus end. So what the cell does is, if it wants something to go in one way, it puts kinesin on it. If it needs it to go in the other direction, it puts dynein on it. And I always shudder to say that, because I say this all the time. This is the way we think about cells, but cells don't think, right? I want this to go in that direction, so I will put this on. It's all about, protein sequences, and it's all spontaneous based on the protein sequences, which is mind-blowing, really. That's what I love about cells. It's all a computer program that plays out perfectly until it doesn't. So anyway, you've got your MTOC here in the center. All the minus ends are there to send something in this direction. You put kinesin on that vesicle. To send something in the other direction, you put dynein on it. So I'd like to show you a little. Let's look at this first. So here, this is how This is that the proteins that connected to the cargo vesicle. So these are just like vesicles in the cell moving things around. So look at this, glycophorin, and here's ankyrin, and here's spectrin, and a short microfilament structure. This is all very similar to the spectrin skeleton that's holding the cytoskeleton to the plasma membrane, which makes sense because this cargo vesicle could end up fusing to the plasma membrane, and then all these proteins are right there, so it just fits right into the plasma membrane. But anyway, here's your dynein. This structure is called dynactin. Dynactin is what holds dynein to the vesicle. And there's dynein. All right, so I want to show you. I showed you a little bit of this on Monday, but I'd like to go back and show you again. Share. Sharing is Karen, right? Window. Oh man. Oh, there it is. Okay. Okay, can you see this inner life of the cell? Anybody?
Yes.
Yes? Okay. So again, this is like 12 years old already. Probably older than that. I don't know. But
We can't see the full thing though. It's kind of like super zoomed in.
Really? Well, anyway, I'm going to play it, let it play, but you know, you can go back and look at it yourself. Mainly what I want to show you here is how the kinesin walks along the microtubule. So here's polymerization of the microtubule. Here's catastrophe. Here's your kinesin. These microtubules all over the place here, coming from that MTOC. All right. That's all I wanted to show you. Now it goes into translation. So let's see if I can get back to something where you can actually see me.
Dr. Slish,
Yes.
could you quickly remind me what the two barrels inside the MTOC are made of?
Yes. I'll go back to that figure. Let me share it. Okay. You see my PowerPoint, right?
Yes.
Okay. Here's your MTOC. This is what the barrel is. 9 of these triplet microtubules in a circle. And they're short, they're interconnected here. That's probably tau or MAP2 anyway, holding them together. Does that make sense?
Yes, sir. Got it. Thank you.
And then the next part that's also important is this paracentriolar material, which is forming your nucleation sites. Okay. All right, so that's cargo movement. So that's one movement we get with microtubules, which is moving vesicles around in the cell as a way of moving proteins around. The other kind of, and here's like a really, really, really ugly neuron, basically. So this is like an axon to the terminal. And you can imagine proteins being made that have to be out here. And so they put your kinesin on a vesicle and send it all the way down the axon till it gets to this end. And now you've got like a myosin attaching it to a microfilament. When you want to send something back, you then put the dynein on the microtubule and drag it back in this direction. So these vesicles have got both motors on it plus myosin, and that allows it to move around, which is pretty cool when you think about it. But don't think about it too much. Just kidding. Okay, so still with our kinesin and dynein, these microtubules are going to be key to mitosis because what happens in G2 before you get into mitosis is that one centriolar pair will duplicate and become 2 centriolars, two MTOCs or 2 centrosomes. As you get into prophase, they will separate. They're going to be connected by microtubules, but they're going to separate and go to opposite poles of the cell. And that is the mitotic spindle. So here, this is basically late anaphase. So no, actually it isn't. It must be like a late prophase or metaphase, because you've got your asters at either side, right? These are your centrosomes or microtubule organizing centers. They're at either side of the cell. You've got microtubules that are overlapping each other, and you've got microtubules attaching to the chromosomes, and you've got microtubules attaching to the cytoskeleton here at the end. So we've got 3 different kinds of microtubules, and they're the same structure, of course. They're all microtubules. We call them 3 different ones because they've got 3 different functions. One microtubule are the astral microtubules. They come off the asters and they stick to the plasma membrane and anchor that centrosome at one end of the cell. Then you've got polar microtubules that are going to overlap with each other on either side from either side of the cell. Connecting them in the middle will be a motor map, okay, a motor that will walk on the microtubules, and this motor is going to walk to the plus side, so that makes it a kinesin. And when this motor walks to the plus side and this one walks to the plus side, it forces those 2 microtubules apart, and that causes elongation of the cell. So you see that elongation in late anaphase as the cell rounds up to go into mitosis. In late anaphase, it starts to stretch, and that's being caused by these polar microtubules. And then you've got the kinetochore microtubules that attach to kinetochore proteins at the center of the sister chromatids. So we'll get into that in detail 2 tests from now when we get into cell division. But for now, just remember you've got kinetochore microtubules that are attaching to the chromosomes. So 3 different functions here for microtubules. Okay, something totally different in terms of movement in microtubules. Now for something completely different. So with microtubules, you also get movement with cilia and flagella. The structure of cilia and flagella are identical. The only difference between them is how long they are. So cilia are really short, and they kind of move in a wave-like fashion. So cilia, of course, if it's an individual cell, it's moving the cell around. What is that, like paramecium have cilia? But in multicellular organisms like us, what the cilia are doing is moving a fluid over the surface. So 2 examples I like to use, because they're the only 2 examples I know, is number 1, in your lungs. Your lungs are constantly secreting a thin, watery mucus, and the cilia in the epithelial cells of your lungs are constantly beating to bring that up out of your lungs. So your lungs are producing this liquid, cilia are bringing it out. And the idea is that when you inhale, if you get bacteria, dust, any kind of crap in your air, it'll stick to that mucus, and then the cilia bring it up, goes down the back of your throat into your stomach, and any bacteria, viruses are probably going to get killed that way. Not all of them, obviously, because we do have crowd diseases, but that's the basic idea. Your lungs are constantly cleaning themselves out that way. The other example, which I've got a video of here, is ovulation. So when an ovary ovulates and a cell comes out, an egg comes out, it goes into that fallopian tube and gets passed down the fallopian tube by this by these cilia that are lining the fallopian tube. So they're forcing fluid down through the tube, and that's bringing the egg cell along. And speaking of the egg cell, along comes Mr. Sperm right behind him, or right in front of her anyway. So the sperm then has got a flagellum, and that flagellum is, again, same structure as cilia, except much longer. So let me see if we can watch this video. I don't know how, again, feeling a little frustrated with my ability to share things on Teams, but let's try this anyway. Bum, ba, ba, bum. Chrome tab window. See, I'm not seeing it now. Chrome tab. Oh, wait. Oh, there it is. Okay. Can you see ovulation?
Yes.
Yes? Okay. So I'm just going to let this play. It's only 2.5 minutes.
Ovulation is a part of the menstrual cycle when the ovary releases a ripe egg, or ovum. Inside the ovary are hundreds of thousands of follicles. Each follicle is a hollow ball of cells with an immature egg in the center. The typical 28-day menstrual cycle begins on the first day of menstrual bleeding. During the first 7 days of the cycle, a few follicles begin to grow at the same time. These maturing follicles secrete estrogen hormone into the bloodstream to prepare the lining of the uterus for pregnancy. Around day 7, all of the follicles stop growing and begin to degenerate, except for one. This dominant follicle continues to grow and nourishes the developing egg inside it. Around day 12, the follicle secretes a large amount of estrogen into the bloodstream. When the estrogen reaches the hypothalamus and the pituitary gland in the brain, the anterior or front part of the pituitary gland releases a huge surge of luteinizing hormone into the bloodstream. Around day 14, luteinizing hormone causes the follicle to undergo a sudden growth spurt. Right before ovulation, the egg detaches from the inside of the follicle. The bulging follicle releases chemicals, causing one of the two fallopian tubes to move in closer and surround the follicle. The follicle swells until it bursts open, ejecting the egg and fluid from the follicle into the abdominal cavity. In response, the fimbriae, tiny projections at the end of the fallopian tube, sweep across the ovulation site and pick up the egg. Microscopic cilia on the
Here we are.
fimbriae's surface transport the egg to the entrance of the fallopian tube. Inside the walls of the fallopian tube, muscular contractions gently push the egg towards the uterus. After ovulation, the egg lives for 12 to 24 hours, so it must be fertilized by a sperm from the male during this time for a woman to become pregnant. If it's not fertilized, the egg dissolves away and is shed along with the uterine lining during menstruation.
Okay, so there is your cilia moving fluid and bringing that cell in. Let's watch Mr. Sperm too. Equal.
Our democracy depends on you.
It really does, folks. That's no joke.
Fluid surrounding them.
So there's the
Next,
fluid.
the sperm will pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates.
Okay. Let's stop our share. Stop sharing. You're oversharing. What is this? Oh, here's another animation I want you to see. Where am I?
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
Okay. So that switch mechanism is not that important for us, but it's kind of cool, isn't it? Like, how does it go back and forth like that? Switch mechanism. But here's
I had a comment about that.
wait. Shut up. Shut up. Okay. So, but let's look at the structure of that
Dr.
structure.
Slice,
Yep.
I just had a comment about that. I wanted to say how I thought it resembled muscle movement, or like how a muscle is triggered to move and like
Right.
flex.
Yeah.
And I was
It's
wondering if
almost
there
like
were any like connections in like literature or like, I don't know. I
That's a
thought that
good-
was really interesting, like how it mirrored it, sort of the way that the microtubules belong.
That's a good question. I don't really know the answer to that if they compare the two. But I see your point, because you kind of have two filaments sliding across each other, just like you do with the actin myosin, and the walking from one side to the other, attached to one, walking on the other. It's very similar, I think.
Yeah, it's just very similar. And the power stroke, it just reminded me of it. I thought it was super interesting.
Yep, absolutely. I would be willing to bet it's calcium based too. I don't know, but I'd be willing to bet that whether, that one side's contract or moving, the other side's not, I'd be willing to bet it's action potential and calcium based as well.
Yeah.
I knew a guy back in grad school in the 90s, 80s, who was doing work with cilia and calcium channels. So I know they are important. All right, so let's look at the structure now. You saw the structure really briefly inside that in that video, but let's look at it in depth here. So here's your microtubule organizing center. When we talk about making axonemal structures, it basically starts with one of the two things. So the cell will make a new centriole, which is a single barrel, and that will go to the plasma membrane and nucleate the production of this axonemal structure. So every one of these cilia or flagella starts in this, and that's called a basal body. It's a single centriole and some proteins that help to nucleate the axonemal structure. And here is your axonemal structure. So in the center, you've got two single microtubules, just kind of structural there, other proteins holding them together. And then you've got these radial spokes that are holding the other microtubules out. So this is all structural, holding it together. And then on the outside, you've got nine doublet microtubules. So you remember back to when we started microtubules, I said they can be in doublets as well. So you've got the A tubule and the B tubule in the doublet. And on the A tubule, you've got attached dynein motors. And this is what we saw in the video. These dynein motors will reach across from the A to the B and grab that B tubule and start walking on it. and that causes these microtubules to slide with respect to each other. And as I'm sorry, I didn't catch your name. As the woman just said, the fellow student just said, this is kind of like a cross bridge, right? It's like cross bridge cycling here. You've got your motor that's reaching across and grabbing this. So, and then another really important part is this nexon that's connecting these tubules. I'll show you why that's important here. So here is those microtubules, the dynein is walking from one to the other and causing them to slide with respect to each other. Again, ATP based. So these dyneins walk and they move each other. Now, what turns that, I mean, if that's all you had, they just move with respect to each other. It might change the length of the flagellum, but it wouldn't whip back and forth. What makes it whip back and forth is the nexon, which makes these interdoubling links. And that, therefore, when one moves with respect to the other, it bends instead of just sliding, you know. So it's kind of like, you know, the inside of a turn, you know, the wheel on the inside of the turn goes slower than the wheel on the outside of the turn. When you got that connection, that translates that sliding motion into a bending motion. So that nexon is really important there. Okay, that's about all I have on microtubules. Do you have any questions on that? All right. Brittany's here. Okay, that's all I have. Can I see your faces so we can be a little bit of a class here for a second? It is 12:04, and I wanted to talk about the exam, which is coming up next week. So remember, we've got a quiz this weekend, which will just be multiple choice questions like the last two quizzes. And I got to be honest, I make these quizzes a little bit on the easy side, so don't expect the test to be as easy as these quizzes. I just want to keep you engaged with the material is the main reason I do that. And then next week from Monday through Wednesday will be the exam. You'll have some multiple choice questions. Some will be the same as the quizzes because I'm too lazy to make up all new ones. But also, you know, you'll have some multiple choice questions which will be worth 50% of the exam. And then the essay questions. I think I'm going to put 5 essay questions on, if that's okay. And these will be a little more complicated. Let me go to share. Chrome tab. Okay, share. Okay, so this is your Brightspace page. This is the way I see it anyway. Just to remind you, all the lectures are up there. When I couldn't get it through Teams, I still did it and posted it in YouTube. So they're all here. Go down below. Don't forget to do this game. This game is mainly chapter 2 because I wrote it a couple of weeks ago. So it doesn't necessarily reflect the later stuff. But but there is Cell Bio Game 1. Excuse me. You can play that game as many times as you want. And then take a screen grab of your favorite score, your best score, and send it to me. Let me run you through that real quick. So here's the game. Basically, you start, and it's just I'm not even looking here. Let's see, Aquaporin. Where's Aquaporin? Aquaporin. Just so I get one right. Let's say so. You just drag and drop, and when you're done, hit check. And these are correct, and these were wrong. So the wrong answer will come up and show you the right answer will come up and show you where it should be. And you've got a timer going here. So if you get at least one right, you can get 1 bonus point for each second left on the clock. If you get them all right, you get 2 bonus points for every second left on the clock. Every question you get right is, I think, 20 points. Every one you get wrong is minus 10 points. So jeopardy attaches, as we say. Okay, and then we've got this. This is probably unfamiliar to you, but I think as you go through it, you'll figure it out. Let's see, glucose transporter, NCX, NKA, calcium ATPase, Sodium. Oh, there's no sodium there. This looks like a proton pump. Where is my proton pump? Sodium glucose transporter, SGLT, and our calcium channel. Watch me rock. I got them all right. I got to teach this class. Okay, so I got 22 seconds left. That'll give me 44 more points. Booyah. Now I'm going to skip the next ones just because I'm lazy. Go through this. And then you got your skeletal muscle. Add bonus. Okay, when you get to your last screen, this tells you how many points you've earned. I got 68 points. I get bupkis. So if you take it seriously, you should easily get in the 800s anyway. And that's 7 bonus points that you add on your first exam score. So don't forget to do that because it both kind of gets you ready for the exam and gives you bonus points. I mean, bonus points, hello. What else? Let me share that again. Share. Back it up. And then here's where your exam 1 will be. There's nothing there now. It's just an empty shell, but that's where it will be. Here is exam 1 practice. So this is typical questions that will be like the last 5, the essay type questions. So you can see what they're like. If you've got, I would suggest looking them over this weekend. If you've got questions about them, let me know. I'll answer your questions. I should probably post the answers to these. Maybe I'll do that today or tomorrow if I've got time. I'll take the exam myself and put the right answers down in a separate file. So anyway, that's the practice for those for the essay type questions, excuse me. But this is where your exam will be from February 5th to 7th. Okay. So that's all I've got for today, unless you've got some questions. No, we all good?
Could you go over, well, I think it's in the Brightspace, but I was just curious about the process regarding, I think we have to get some external program to take the exam and whatnot.
Yes, I'm new at this. I don't really have all the details, but I will send you an announcement from Brightspace with all the details of that. But I haven't figured it out myself yet. So I'll figure that out today or tomorrow, and then I'll send you an announcement on that. Okay. It's like a browser lockdown. So when you're taking the exam, you can't, scan the internet and look for answers and stuff. It's kind of a thing. Okay. Any other questions? Okay, well, let's wrap it up. It's been lovely talking to you. Hope we can do this again. So next week we won't meet on Monday because you've got the exam. So that'll be your exam time. So the next time we meet will be next Wednesday, and we'll start chapter 8. Okay. All right, guys. Have a great weekend. Again, you have any questions, just give me a yell. Email is the best, and I will attempt to answer your questions. I know most of this material, maybe not all of it. All right, later, guys.
Thank you.
You're welcome.